Thank you for joining us for the first session of our two-part webinar series with Dr. Jason Ackerson. My name is Shannon Muller, and I'm a soil health educator with the Soil Health Institute. My colleagues, Odine Alvarez with the Soil Health Institute and Olivia Cayuette with the University of Missouri's Center for Regenerative Agriculture, are also on the call providing technical support. We three support the education component of Farmers for Soil Health. The purpose of today's webinar is to provide you all with an overview of SHI's recommended soil health measurements and discuss how they can support efficient, scalable soil health sampling efforts. Today's webinar is supported by Farmers for Soil Health, or FSH. This initiative is one of many USDA-funded Advancing Markets for Producers projects. FSH is a collaborative initiative of commodity organizations, the United Soybean Board, the National Corn Growers Association, and the National Pork Board, with a goal to increase cover crop adoption. This specific project provides an incentive to farmers who are new to planting cover crops or who have been planting cover crops for many years. State-level commodity organizations and conservation professionals work across 20 states, stretching from the Dakotas to the East Coast, to provide local technical assistance and outreach programming. FSH has also developed a website full of resources and information. You can learn more about FSH at farmersforsoilhealth.com. Lastly, a few items about today's webinar. This presentation is being recorded, and an email will be sent with a link to the webinar recording posted to the Soil Health Institute YouTube channel, along with a final feedback survey. We'll end today with a Q&A moderated by Odine. So, as questions come up, please put them in the Q&A. You can upvote questions that you'd like to prioritize as we cover them live. If you run into any technical issues, please don't hesitate to chat the webinar co-hosts, and we will work to resolve any issues. If you'd like captions on, those can be enabled by clicking the three dots on your toolbar and navigating to captions. Be sure to hang on till the end for information related to the Certified Crop Advisor Continuing Education Unit for attending this webinar today. With that being said, I am excited to welcome our guest and a colleague of mine, Dr. Jason Ackerson. I'll let Jason introduce himself and take it away.
Thanks, Shannon, for that really thorough and welcoming introduction. I'm happy to be here with everyone today, speaking about some of the work we've been conducting at the Soil Health Institute over the last several years, thinking about the process of measuring soil health and benchmarking soil health, really to give growers and their advisors the tools they need to sort of answer the question like, how healthy is my soil and how healthy it could become? So as Shannon mentioned, this is part one of a two-part webinar series outlining our measurement philosophy and benchmarking approach for quantifying soil health. So the first part of this webinar, what we'll focus on today, is really on the core components related to what do we talk about when we're talking about measurements of soil health. And really what I want to hopefully share with you today is the story of how the Soil Health Institute, came about our essential soil health indicators and the scientific work that went behind the scenes to really crystallize around what we're talking about when we're talking about measurements for soil health. And then in our next webinar series, I'll share with you sort of our approach for taking those measurements that we're going to describe today and really actualizing them in a system to help answer the question, how healthy is my soil now and how healthy it could become relative to, similar soils in my geography. So that will really be focused on, the approach of understanding, regional soil health potential and how to interpret those values in the context of reporting to growers, but then also understanding, the inventory of soil health at a regional scale. So for those of you who are unfamiliar with the Soil Health Institute, I'll give you kind of a quick background on who we are. Soil Health Institute is a global nonprofit with the mission of safeguarding and enhancing the vitality and productivity of soils through research and enhancement. That's just a really wordy way of saying we're a bunch of soil scientists and agricultural professionals who really care deeply about the health of our soils and making sure that soil health is part of the solution for providing sustainable and productive agriculture in North America and beyond. And a lot of the work and mission around Soil Health Institute for the last 5 or 6 years has really been focused on this question around measuring and quantifying soil health. But maybe stepping back a little bit, I want to talk briefly about why soil health matters and why we really care about it. And I think if you've seen a Soil Health Institute talk, you've probably seen this exact slide before, but I think it's really illustrative of why we all care so deeply about soil health and the management of soils. And that really is thinking about ways of improving soil function, environmentally, ecologically, and most importantly, agronomically. So what you're looking at here on the slide are two soils split across the fence row. On the right-hand side, you're seeing a soil that's been managed for soil health. You'll see lots of residue cover. We're minimizing the soil disturbance. We're maximizing the amount of residue cover, and we're doing things to promote the continuous living roots in that soil. So we've done everything we can to promote the good functioning of that soil. On the left-hand side of the screen, we're seeing growers who aren't doing the same practices, right? You won't see a lot of residue cover. There's, probably a certain high degree of soil disturbance through tillage, and there's lack of living roots throughout the season. And that has significant impacts on the function of the soil in that system. You see lots of water ponding on the surface. There's lack of infiltration, and that has impacts on, both the environmental consequences of that, increased runoff, increased erosion, but also the agronomic consequences. So you'll see, issues with poor stand emergence, potential loss of yield, and really, important consequences both for, off-site stakeholders, but maybe more importantly, the growers on the field, the farmers who are trying to manage these soils effectively. And I think that concept and that focus on soil function is really critical for us to understand the story around how we measure and quantify soil health, right? So soils provide a ton of important ecosystem services and functions, like I mentioned, on and off the farm, right? They're integral to water infiltration and storage. Obviously, they're a central component of crop production, and grain and fiber production. They're also related to things that interact with global climate regulation and the cycling of carbon and organic matter in systems, right? Soils are a fundamental part of those global biogeochemical cycles. And of course, they're certainly really important vehicles for nutrient cycling and storage that directly relates to crop production and agronomic outcomes on the farm. So having healthy and functional soils really is important to providing all of these services. And when we think about the interaction between soil function and soil health, really it's where we see the sort of two components of soil properties kind of merging up. So the first is, what I would call inherent soil properties. And we'll talk about this more in the coming webinar around benchmarking, but really, soils inherit a set of constraints or conditions that are unaffected by management to a large degree. These would be things like the soil's natural drainage class, the soil texture, the kind of climate that soil was formed in. That really provides the sort of like initial context for understanding the constraints around that soil's function. Right? A really clayey soil is never going to have the infiltration rates of a really sandy soil, no matter how they're managed, right? There's a limit on the physical expression of those functions. But there's actually quite a bit we can do on the management side to either improve or degrade those functions, right? We can adopt soil health management practices, things like cover crops, reduce tillage, promoting good soil armor and soil coverage that can enhance those soil functions and promote good ecosystem services on the soils. And when we combine that sort of inherent set of properties with the management context, we end up with, the set of soil health parameters that describe, the current state of the soil as it relates to those environmental functions. So thinking about soil health is the expression of those inherent properties and then the management of those soils, we get to soil health. And I think this is where when we ask the question, why would we measure soil health? It really is because it's an expression of those two components. So soil health measurements can help, growers identify potential problems with soil functions. They can be used as part of a conservation planning assessment. And we'll talk about how the NRCS is using soil health testing as part of that in emerging programs. And of course, managing and measuring your soil health is part of tracking what happens as we change practices, right? If a grower adopts a conservation practice, it's really, helpful for us to understand how that practice is changing the soil over time. Um, and lastly, our CSO, Christine Morgan, likes to say, if you treasure it, you measure it. I care deeply about soils. I want to know that they're healthy. And so I think there's also some inherent value in us understanding how healthy a soil can become. Um, and so, understanding, why we might care about measuring soil health doesn't necessarily get us to an answer to that question of how do we measure soil health? And this has been one of the big projects of the Soil Health Institute since its inception, is thinking about, when we take all of the potential things we could measure around soils, what are the best indicators of soil health? And to kind of unpack that really tricky question, the Soil Health Institute led a project called the North American Project to Evaluate Soil Health Measurements. You'll often hear SHI folks refer to this as NAPSHM, which is kind of, lending in the long tradition of the USDA, one of these terrible acronyms we see thrown about. Um, but essentially, the goal of NAPSHM was to identify what are the best measurements or indicators of soil health. So SHI staff went to 124 long-term experimental sites across North America. So these are places where we have replicated trials of management practices that change soil health. These could be long-term tillage trials, they could be long-term cover crop trials, they could be trials on, crop rotations or biological amendments. Really anything that was replicated at an experimental station was considered as part of this trial. And importantly, right, because these are all at long-term experimental sites, places run by the USDA ARS or land grant institutions or similar NGOs, we have tight controls over what those experiments look like. We have replication. So we have that good sort of scientific experimental basis to use in our inquiry. So at each of those sites, SHI scientists went out and collected soil samples from 0 to 15 centimeters from all of the paired treatments at those sites. And at each of those samples, we measured 30 different measurements of soil health. And so I'm going to do the classic scientist thing where I'm going to show you a big giant table and I'm going to say oh, don't worry about all the details in the table. Really what I want you to understand is this is a really exhaustive suite of measurements. We focused on basically if you could measure it or with regard to soil health, we tried it in NAPSHM. And really the whole goal of that was to think about from this suite of measurements, how do we pick the ones that we think are the best things to move forward with the process of measuring soil health. And to maybe unpack that table a little bit better, we can kind of take some of these indicators and break them up into different buckets, right? So we have some measurements that are related to the biology of the soil. So these would be things that describe the carbon pools or the carbon cycling dynamics or the microbial expressions of soils. We have measurements that relate to the sort of physical properties of the soils. Some of these are going to be inherent properties like texture, but there are also these sort of dynamic physical properties, aggregate stability, infiltration rates, available water holding capacity, all of the things that regulate the physical, mass and energy transport within those soils. And then lastly, there's a group of these parameters that are really related to the soil chemistry. These might be things related to the electrical conductivity, the pH, the nitrogen and phosphorus, the kinds of nitty-gritty things that soil chemists like to think about. And when we think about where all of these interrelated measurements kind of intersect, that center of that Venn diagram really is what we're thinking about when we're thinking about soil health. These are measurements that are sensitive to the biological, chemical, and physical components of soils. So how do we take all of this complicated, messy measurements, that big table I showed you, and refine it into a set of measurements? So We applied a series of filters to those nation data. And it's sort of, you could think about it as increasing levels of specificity. So at the first level, we went through all of the available literature. And we picked out the measurements that we thought were reflective of soil health, and therefore we removed anything that wasn't from the analysis. And then we went into the actual results from the NAPE and we asked the question, okay, which of these indicators changes when we change the management of the soil? If, for example, adopting no-till for 30 years doesn't change that indicator, it's probably not a very good indicator of soil health. So we removed some of those measurements that weren't very sensitive or sensitive across large geographies from our study. So, some measurements were responsive at some sites, but not at every site, and therefore they were excluded from our future analysis. Lastly, we thought about um, both scale and the redundancy of measurements. So we wanted measurements that were widely available for growers that could be conducted at commercial labs. Some of these measurements are really great for scientific research, but they're not scalable because a commercial lab can't run them, and therefore they're unavailable to the average grower who might be interested in measuring their soil health. Um, and then finally, thinking about we wanted to minimize the redundancy of measurements. So if two measurements were similarly sensitive to management and also kind of both represented similar portions of that physical, chemical, or or biological space, we tried to pick one winner from those groups. So the first filter, we removed a lot of measurements that were really not reflective of soil health. So the standard fertility measurements, um, NPK, micronutrients were removed. We removed measurements that were essentially measuring the inherent soil properties like texture, and then some of the more, scientific oriented measurements that are a bit more related to, further scientific discovery were removed as well. Then the next filter that I mentioned was thinking about measurements that were responsive to management. So the graph on the right here is showing, a snapshot of one of those measurements. For example, this is soil organic carbon. And what you're looking at here are the response rates of organic carbon to the various treatments in NAPESHM. So these data here are pooled across all of the NAPESHM trials. And for each of those, horizontal divisions relating to a management practice that was in the NAPESHM trial. So rotational diversity, number of crops, residue retention, you'll see that aligns well with the soil health principle of keeping the soil covered, organic nutrients, cover crops, again, increasing living roots, another soil health practice, and then decreasing tillage, which is minimizing soil disturbance. And when you look at that vertical line, which represents zero or no change in organic matter, any one of those boxes or dots that intersects that line means that across all of the NAPESHM data, that indicator was not sensitive to management. So for example, here we're looking at soil organic carbon, and in fact, for all of the sort of soil health principles, organic amendments, residue retention, cover crops, and decreased tillage, we see that the line is away from that zero line, which indicates that that measurement, soil organic carbon, was sensitive to those management changes across NAPESHM. So a really good indicator of a measurement that is sensitive to management. So anything where all of those dots, any of the measurements where all those dots would intersect that zero line would have been excluded from the second stage filter. And then our next filter was, applicable at scale. Some measurements, things like infiltration rate, my background's in soil physics. I would love to see every grower go out and measure, hydraulic conductivity on their fields, but that's not an applicable or scalable measurement. It takes too much time, it's too expensive, it's too labor intensive. So measurements like that were excluded from our analysis. Another great indicator is available water holding capacity on intact cores, super predictive of a lot of great outcomes. It's directly related to soil functions, but there aren't a lot of labs that are going to take an intact soil core and then do the work to measure available water holding capacity. So that measurement really doesn't scale well because it's not commercially available to growers. And then our last filter was thinking about making sure we minimized redundancy in our analysis. So there are a lot of indicators where they're measuring really similar things. So we measured multiple measurements of aggregate stability that all indicate the same physical process in the soil that relates to soil structure. We don't need to measure three different measurements of aggregate stability. What we need to do is pick one that's most widely available. And so that's sort of the last filter we went through is once we knew everything that was sensitive to management, everything that was commercially available or scalable, and then not redundant, we ended up with what we call SHI's essential soil health indicators. So the first indicator we'll talk about is soil organic carbon, which our method of what we approve is measured through dry combustion. And this really describes the chemical structure of the soil, how much carbon is there as the basis of the microbial food chain. Our next indicator is carbon mineralization potential, which we measure through a 24-hour CO2 burst method. And this really describes the biological structure of the soil. And then our last two measurements relate to the sort of physical properties of the soil, aggregate stability and plant available water, both measure different portions of that soil physical continuum. And so you'll see we ended up with from 30 measurements, we winnowed it down to really three measurements and four total indicators. Um, and each of those is reflective of a different part of that soil health, worldview. One chemical indicator, one biological indicator, and one physical indicator. So what we're going to talk about for the remaining time is kind of go through each of these essential indicators one by one, talk through some of the complexities and nuances about them, how they're measured, and then we'll wrap up with kind of a preview of the benchmarking approach. So, I mentioned earlier that one of our filters was picking indicators that were responsive to management. So you've seen a similar graph like this before. This is showing the responses of our indicators to the different treatments in NaPCM. So we've kind of flipped the axes, but on this example, each one of these graphs represents a different management treatment in NaPCM. So we have organic nutrient sources, application of cover crops, reductions in tillage, and increased residue retention. So each of these align with our soil health principles. And then vertically, each of those dots represents the different essential indicators for soil health. So we have carbon mineralization potential, that's our biological indicator. We have aggregate stability, that's our physical indicator, and we have organic carbon, that's our sort of chemical indicator of soil health. And you'll notice that for the majority of these indicators, with the exception of aggregate stability for organic nutrient sources, those dots and lines are to the right of that vertical line. So if you remember, that vertical line represents that there's no difference in that indicator for our paired treatment in NaPCM. So said another way, when all of those indicators are to the right of that line, it's telling us that those indicators are sensitive to that management practice. So when we have, for example, cover crops, our carbon mineralization potential increases, it moves to the right. When we have when we have cover crops, that aggregate stability increases, it moves to the right. When we have cover crops, our organic carbon increases, it moves to the right. So when we look at our suite across all of the data from NaPCM, we're seeing that each of those indicators is sensitive to soil health management. So they're really good at telling us that the soil is changing when we adopt a soil health management practice. So going into our indicators, soil organic carbon, like I mentioned, it's the sort of chemical indicator of our group. And I like to think about it in probably our soil microbiologist on staff would hate this analogy, but I think about it as like fuel or food. It's the basis of that microbiological food chain in soils that helps do all the important things that microbes do. Um, and it's composed, organic carbon is composed largely of non-living organic matter, crop residues, or root exudates. And we measure it through a process called dry combustion, which gives us, the total amount of carbon in a soil in arid and semi-arid regions where we also have inorganic carbon, carbonates in the soils. We account for that through subtraction of inorganic carbon. And our total soil organic carbon is the total amount of carbon minus any of those carbonate carbon molecules that are related to non-organic processes, precipitation of calcium salts in the soil. So a lot of growers are familiar with organic matter, and it's a really similar indicator to organic carbon. They're slightly different in the way we measure them. The reason, we really like soil organic carbon rather than organic matter is because it's less sensitive to some of the issues related to soil texture. So on this graph, we're seeing carbon as measured through dry combustion versus carbon as estimated on loss and ignition organic matter. And you'll notice that the loss on ignition data tends to fall above that solid line. If the data were completely interoperable, they were interchangeable, that all of those points would line up along that one-to-one line, meaning that one atom of carbon measured with dry combustion is equivalent to one atom of carbon measured in loss on ignition. But you'll see tends to be a positive bias in that loss on ignition carbon. And maybe more importantly, is that that bias is related to the clay content or the texture of the soil. So the more clay in the soil, the more biased that number becomes. So the more we're overestimating the amount of carbon with loss on ignition. So this is one of the reasons why dry combustion is a lot more precise of an indicator, is that it doesn't have this textural effect that we see with loss on ignition carbon. So, one of the reasons we approach carbon is this lack of textural effects. We'll also think about carbon as it relates to soil carbon stocks. So anyone who is in the soil carbon trading marketplace or thinking about soil carbon credits has probably heard the term carbon stock. In order to get that measurement, we need the soil organic carbon concentration that we measure as our essential indicator, but we also need a measurement of the bulk density, and typically we would measure those data down to 30 centimeters. So you need a little bit more data to estimate a carbon stock, but the central component of that carbon stock is our essential indicator, soil organic carbon. And then similarly, you'll notice that we don't have any indicators that explicitly talk about nitrogen in our essential soil health indicators. And that's really because carbon and nitrogen are so tightly coupled in biogeochemical signatures. So what you're looking at here are NAPSIM data presenting a carbon indicator plotted on the x-axis and a nitrogen indicator plotted on the y-axis. So we have soil organic carbon through dry combustion and organic nitrogen through combustion versus water available carbon and water available organic nitrogen on the graph on the right. And in each of those cases, you'll see a really, really high correlation with really high R squareds. 0.92 is very high in soil science. That says, organic carbon and nitrogen are very, very tightly related. So when we talk about, minimizing the redundancy of our measurements, this is a good, expression of that if we know the carbon content concentration, we have a pretty good idea of what the nitrogen concentration is going to be. Therefore, we don't really need to measure nitrogen as an additional measurement. We can get by with just understanding the carbon concentration. Our next indicator that we recommend is carbon mineralization potential. This is a really good measurement of the soil's biology or the biological activity of a soil from the soil microbiology. We often hear this indicator referred to as a CO2 burst or soil respiration, and that's really because that's what it's trying to measure. It's measuring the CO2 or the amount of respiration that's happening. And it really is an indicator of the soil's response to the soil microbiology response to re-wetting. And as those microbes, metabolize and do their microbe thing, right? They're cycling nutrients, they're doing everything. They're respiring and emitting CO2. And so we can measure that CO2, and the amount of CO2 we measure is related to the microbiological activity of the soil. So more CO2 indicates a higher amount of microbe activity. The way we measure this in the lab is on dried ground soils. So similar to the kind of processes we would prep a soil for standard fertility analysis, soil gets dried and ground, and then we put it in an airtight container and re-wet it. And those samples are let to sit for 24 hours, and we measure how much CO2 evolves off those samples in that 24-hour period. So by quantifying how much CO2 comes off, we get a pretty good indicator of how much the microbes are respiring during that 24-hour period. Um, I think this is important to talk about, but like the unit we use here often is in milligrams of carbon per kilogram of soil per day. So kind of an abstract unit if you're not into the sort of scientific nomenclature, but you can really think about it in terms of the amount of carbon, the mass of carbon that's coming off per mass of soil over that defined period. So our next measurement, this is a physical indicator of soil structure, is aggregate stability. And I mentioned that, this is our physical indicator and it really describes the resistance of the soil fabric to falling apart or, how well it stays together under wet conditions. It's a good indicator of soil structure. So higher aggregate stability is associated with better expression of soil structure, which leads to higher macroporosity, and then all of the associated physical outcomes that come with good soil structure, better infiltration rates, resistance to erosion, both wind and water erosion. So those physical soil functions are really strongly linked to soil aggregate stability. There's a lot of ways to measure aggregate stability. Our preferred method is aggregate stability through image analysis. This is something we're helping commercial labs get stood up with. But essentially, if you've seen the sort of slaking demo at a field day where someone takes a soil clod and puts it into a jar and lets it fall apart, this is a quantitative version of that same process. So either using a camera or our smartphone application, which I'll talk about on the next slide, we place an aggregate in water. That's what you're seeing on the bottom left-hand image. And over time, the water gets absorbed into that aggregate and it falls apart. And we can actually measure how much that aggregate is sort of sloughing or slaking or falling apart over time. And that gives us our indicator of aggregate stability. So more stable aggregates will stay together and be more intact and be less dispersed at the end of that slaking process. So one of the great things that I think that SHI has done recently is develop the Slake smartphone app. So hopefully some of you have seen this, but if you haven't, it's a free app that helps you measure aggregate stability. I put up some QR codes here, but we'll also provide a link in the chat for where to access this app. Um, like I said, it's a free application for Android and for Apple that helps you do this aggregate stability measurement on a smartphone. So it employs all of the same procedures and methodologies. You place an aggregate in a petri dish or in some sort of container, take a photo of it with the application. It instructs you on what to do next. You add some water, and over time, it takes a second image and then can compute the aggregate stability on your smartphone. So a really useful tool if you're, doing demos at conservation field days or on farm with growers, but also, if you're a grower who wants to track your aggregate stability, this app is a good way to do that. Um, I won't get into a lot of the details. We have more resources on our site that kind of walk you through aggregate stability with slakes. Um, and then our last sort of measurement or indicator of soil health is available water holding capacity. This is a physical measurement that describes how much water is stored per volume of soil. We estimate this using a pedotransfer function. So if we know the soil's texture and we know its organic carbon concentration, we can predict how much water is stored in that soil. And those data were all derived from the NAPSHM project. Um, but essentially, what this helps us do is say if your organic carbon content concentration is of a certain percentage, based on your texture, we can tell you how much water is held in that soil. So the graph on the right is an example of one of the outputs of this pedotransfer function. So you'll see, we have four lines representing various levels of organic carbon concentration. So at the top, we have 4% going all the way down to 1% at that bottom line. And then on the x-axis is the texture or the clay percentage of those soils. So for any given texture or clay content, as we add organic carbon to that soil, we see an increase in the amount of water that's stored in that soil. So when a farmer adopts a conservation practice that promotes the storage or sequestration of carbon in that system, our pedotransfer function predicts the predictive incremental change in the water storage for that soil for that carbon concentration. You can also measure available water holding capacity using, direct measurements, and certainly those are available. But again, coming back to perspective of trying to find measurements that are scalable and commercially available. Um, there aren't a whole lot of labs that will run available water holding capacity on an intact core. So this pedotransfer function way is really the simplest approach to estimating organic carbon, or excuse me, available water holding capacity. Um, Lastly, I'm going to touch really briefly on a new federal cost share. So we talked about SHI's essential soil health measurements or indicators. There's a new, not a new program, the Conservation Evaluation and Monitoring Activity, SEMA 216, has been available for several years now. But it's gaining a little bit more visibility with the launch of the USDA's Regenerative Pilot Program, which is the new unified application for EQIP and CSP. So um, both of those programs now have a shared application. And as part of that RPP program, SEMA 216 is a required practice. Um, so this is a soil testing procedure where a qualified individual, in consultation with a farmer and NRCS staff, goes out and performs a soil health test for that field as part of a conservation evaluation and monitoring activity. And I just want to note that SHI's essential soil health indicators are included in the basic soil health test in SEMA 216. So if you're doing cost share, it can cover the cost of our measurements or our indicators. Lastly, just to kind of wrap up, I know there's a lot of information we presented on a lot of technical things. Um, SHI has a ton of resources available on our website. We have a whole landing page dedicated to measurements of scaling soil health assessments. So this is all of the details from the results of the NAPSHM procedures. So we have standard operating procedures for each of our measurements up there. We have links to all of our peer-reviewed publications that really show you, help show the work on how we got to this essential set of soil health indicators. So those are all linked on the website, as well as some plain language fact sheets and summaries of all of the information I presented today on our soil health indicators. We also have a landing page specifically to our Slake smartphone app that measures aggregate stability via image analysis. So there's a fact sheet there that walks you through how to use Slakes. There are also links to both the Android and Apple web stores where you can download the application for free. Those are all also linked on that website. So both of those are great resources to go through and pick up more information if you are interested. So just to kind of wrap up, we can measure soil health scalably and scientifically rigorously with a pretty small set of soil health measurements. And, a lot of work at the Soil Health Institute has gone into refining our approach to measuring these indicators. And that leads us to, SHI's essential soil health indicators of soil organic carbon, carbon mineralization potential, and wet aggregate stability as the measured components of that. And when we pair those with the pedotransfer function for available water holding capacity, we have a pretty robust set of measurements that can be conducted, across large spatial extents and with a pretty limited set of investments that are available at commercial labs. So this is a commercially viable approach to measuring soil health. So it's more cost effective and it really provides the key information you need to make management decisions or track, monitor soil health progress. If you're interested in doing soil health testing, there is a federal cost share available that covers some of the cost of that activity through SEMA 216, and SHI soil health indicators are covered by that SEMA 216 cost share. So really useful if you're part of that RPP program, you can utilize SHI's essential indicators for that activity. Um, and we talked a lot today about measurements. I will talk later, March 5th. We'll have a follow-up seminar where we'll talk about how do you take these measurements and utilize them in an analysis framework that we call soil health benchmarking, which really helps you understand or answer the question, how healthy could my soil be and how healthy are similar soils in my region? So stay tuned for that. We'll talk more about that in the coming weeks. So I think Shannon, Odine, Olivia, do we have time for some questions?
Yes, we do. And yeah, well, first of all, thank you so much for the great presentation, Jason. It was really helpful to understand this story of how the Soil Health Institute does their measurements and quantifies soil health. So that was really helpful, and also understanding how you picked the best indicators of soil health. So we're gonna dive into the Q&A, and we've received several questions already. And if you have more questions, you can continue to put them in the Q&A. Let me get. So the first question was about the Naption research, and it's a little specific. They're asking why was 0 to 15 centimeters depth chosen, and if there's a larger data set, like maybe more depth, maybe that's what they're referring to.
Yeah, so we honed in on that 0 to 15 centimeters, partially based on the literature, right? A lot of these indicators were really initially tested and developed for that surface bit of soil. And we see in the literature that when a practice is responsive to changes, it's most commonly or when a measurement is responsive to a change in management. And
Mhm.
most commonly, that change in measurement happens in that 0 to 15 centimeter region. It also aligns very nicely with our sort of existing infrastructure for soil testing. Fertility testing often focuses on just that surface layer of soil. So it's another way of thinking about the scalability of these measurements is having those measurements compatible with, our existing testing infrastructure.
Mhm.
In the carbon space, there's a lot of interest now in thinking about measuring carbon stocks. And again, for a total accounting of the carbon stocks, you certainly want to go deeper than that 15 centimeter depth. We
Okay.
recommend at least going to 30 centimeters, but there's a lot of interest now from certain carbon markets or registries of thinking about going even deeper to account for the total amount of carbon in the soil. It's just that 0 to 15 centimeters is where we're most likely to see a change in soil health in response to management. So that's why we focused on that. I
Okay.
will note, Olivia, or sorry, Odine, that the NAPTION data are available if people are interested. Those data are publicly accessible for most of our sites. You can drill down into those data in more detail. And there's a suite of publications that are linked on our website, going into NAPTION in a lot more detail. So,
Uh-huh. Yeah, that makes me think that maybe we can include some of those resources, like the publications, when we send that email with the
and
breakdown.
I'm sure all of the SHI scientists would love as many people to read those papers as possible.
Okay. Okay, so we'll make sure to include those in there. And there was a question related to measurements of carbon. Let me see if I can find it. Yeah, since we're in that topic. So
Yeah.
someone asked, how can carbon credits be quantified using different soil carbon measurement methods, such as the Walkley-Black method?
Yeah. So, I'll first say this by, our essential soil health indicators are focused on like a holistic perspective on measuring soil health. So we try not to, in the context of understanding soil health, I think it's important to not just focus in on carbon. That's just why we include, carbon mineralization potential and aggregate stability in our essential soil health indicators. But, with regards to other measurements of measuring carbon, um, for carbon crediting specifically, there are criteria for how we actually measure those credits.
Right.
And they're described and standardized by whatever the registering body is that's certifying those carbon credits. So the two leading registries would be Vera and CAR. Each of them have their own criteria for developing carbon accounting schemes. Um, but in at least in car, I believe the standard procedure that's recommended is is dry combustion for carbon. So Walkley Black might not be considered adequate, but I would defer to those car and Vera standards for how they're actually developed. I mean, the whole world of carbon crediting and carbon accounting is, the measurements are such a small portion of actually generating a credit. Then there's, how do you wrap that up into a project? Um, and we're seeing, a lot of complexity there. So I would defer anybody who's interested in that to reaching out to somebody who's actually involved in that carbon accounting space from a project developer or design perspective to better understand how their data could go into a carbon crediting scheme. I
Mhm.
know there are probably some exceptions for new or different carbon measurement technologies, but that stuff is all, emerging right now. So that's
Okay.
probably a not a very specific answer to that, but
It-
and like everything with carbon credits, it's complicated.
At least it guides the person to where they need to go and look for that information. So thank you for that. We actually got the same question two times on why does your soil microbiologist on staff does not like the soil organic carbon um, as a fuel analogy? Yeah.
Oh, I
Yeah.
don't think that's true. I think that's just a gross oversimplification and me taking
Okay.
pot shots at a scientist
Yeah.
who's not here to defend
So
himself.
would you think is like a good analogy to use like to explain in education settings?
Yeah,
Like I want to
I think
explain
it
it
is.
in
That's
this.
why I use it here, right? Is I think it's a nice
Mhm.
simplified approach. I think
Okay.
there are a lot of, anybody who spends time around the detailed science around how carbon and microbes interact in the soil understands how like that analogy is very a very simplified
Simplified.
approach.
Hmm.
Um, but it does a good job of describing most of the dynamics there, but there are also a lot of complicated interactions that are so straightforward, right? When we think
Hmm.
about the organic carbon concentration of a soil sample, it includes, all of the necromass, all of the dead carbon, but also any of the microbial carbon that's, part of existing living organisms. And so when we think about the carbon concentration as just the fuel for microorganisms, that's a little simplified because it also includes the microorganisms themselves. So,
Oh,
there's a lot of nuances there. So,
okay.
I will defer any specific questions about soil microbe and carbon interactions to our microbiologist, Liz Reiki. So send all your inquiries and questions to Liz at Soil Health Institute. So
Okay.
happy to punt that to someone else. But yeah, no,
Yeah.
I think it's a great like starting point analogy, but
Hmm.
like all analogies, it does sort of simplify maybe oversimplify a complex
Processes.
set of
Hmm.
dynamics. So as a starting point, I think that's why I use it. It's a good way to think about it initially, but recognize like everything in science, particularly in natural sciences, like when you start getting into the details, it's way more complicated than we could ever describe in a one sentence soundbite, which is why
Yeah.
it took me 30 seconds to answer that question.
Well, thank you for taking the time to explain. And hopefully that answered the question for them. We have several interesting questions, but I want to go to this one. Which labs are or will be trained for the soil health assessments? That's the
Yeah.
first one.
Um,
Like testing for
so
SEMA 216, like the soil health indicators. Yeah.
yep. Okay,
Mhm.
so I will speak to SEMA more broadly from the starting point, and then I'll talk about our indicators specifically. Maybe briefly. Any laboratory can provide SEMA 216 compliant soil health indicators, provided they're doing measurements listed in SEMA 216, which include our indicators, but there are also alternative measurements for those same indicators that are available. So, for example, like I mentioned, there are multiple measurements of aggregate stability and they all sort of can qualify for SEMA 216 funding. So you don't have to go to a lab that does image analysis in order for it to be compliant with SEMA 216. What you do need for SEMA 216 is that lab is certified through the standard testing proficiency program or other laboratory proficiency. I think they provide both um, lab testing programs are sufficient criteria for labs, but I would defer anyone to the SEMA 216 documentation before committing to that. Part of the SEMA process is that the qualified individuals and the farmers and NRCS representatives have a pre-work meeting where they can go through all of the specifics of SEMA. And one of the reasons for that meeting is to make sure that any laboratory that they're sending samples to would qualify as a SEMA compliant lab. Um, and then, the Soil Health Institute, I haven't, didn't mention this much in our talk, but we are working with several labs to, help them come up to speed on our soil health indicators. So if you are looking for a laboratory, reach out to someone at the Soil Health Institute and they can probably provide you with a guidance on how to select a laboratory that is using our indices. If you are a laboratory and you want to come up to speed on SHI's essential soil health indicators, feel free again to reach out to us. But probably the first best place is that um, link to our measurements landing page that includes all of our standard operating procedures and
Mhm.
troubleshooting methods. So
Okay.
that's a great place to start if you're a laboratory working in this space. But always, reach out to us and we can help try to
Yeah.
guide you in the right direction.
There's your email, and we'll put our emails too at the
Yeah.
end. And there is a related question about what is the difference between a regular soil test and a soil health test. And I think that relates to other question about why you filtered out fertility metrics of NPK as a measurement of soil health, like that you showed that in your presentation.
Yeah. So, you know, I'll start by saying, from the sampling perspective, right, we can do a soil health test in a lot of the similar ways that we would collect a soil fertility test. So the actual process of collecting samples for soil fertility and soil health aren't that dissimilar. There are some special considerations for handling samples. Again, I would defer people to our standard operating procedure for sampling, which is on that website that we provided the link to in the chat. So if you want more details, again, I'm probably sound like a broken record, but I think there's too many details for me to cover in just a Q&A session here.
Mhm.
But backing maybe to the question more specifically around, why when we talk about soil health, we're not talking about soil fertility. And I think, you know, one of those is philosophy, right? We already have a robust system to manage soil fertility as irrespective of soil health. So, we could add soil fertility testing to an existing soil health test, but they're complementary measurements and sets of diagnostic pieces. They tell you slightly different things about soil health. I think it would be I would not want to see growers ignore soil fertility in favor of soil health testing. I don't think they are antagonistic measurement approaches, but I think they're complementary. And that's why we didn't say, soil health testing has to have fertility testing in it. Partially, a lot of growers are already conducting soil fertility testing, and largely, soil health is complementary to that. So I think, it's not that soil health and soil fertility aren't very related, right? We know that increasing soil health has effects on carbon and nutrient cycling in soils. That's one of the functions we're trying to manage with soil health management. Um, but they're not perfect overlaps. So I would say they kind of overlap on a Venn diagram. So when we're thinking about just things that are sensitive to management, things like the potassium concentration isn't going to change significantly with something like adoption of no-till. But so it's also not a great, the fertility measurements themselves aren't very sensitive to management in the way we want a soil health measurement to be. So I would advise anybody who's approaching soil health measurement or testing to also consider the fertility component of their soils. It's really hard to grow cover crops if your soil fertility is really poor, right? If your pH is not managed appropriately, you're going to struggle in maintaining good soil function. So I think they're complementary components of understanding the whole picture of soils and how to manage them effectively.
Thank you. That's such a helpful explanation. And one question that is intriguing is, do you think there are important functions you're missing with this suite of measurements where you think you need to develop a better measurement in the future?
Ooh, That's a really great question.
Maybe
I
you
think there's a lot of exciting work in soil
Mhm.
science right now and thinking about how other things we can measure. Um, and other functions that might be really important. So, one of the things we measured in NaPSHM that is really, I would say on the cutting edge is any of the microbiological genetic approaches, right? So we measured different types of sequencing, shotgun metagenomics, the 16S sequencing. So we have a lot of, biological like um, genetic profiling of these soils. So I think, there's a significant interest in the scientific community in understanding, how microbial communities themselves change in response to management and how we measure that. So I think that's one place where we address it in a kind of roundabout way by measuring the, biological activity with carbon mineralization potential, but we're not measuring the actual, community structure of those microorganisms. So if there's one place where I think there's a lot of room for improvement in measuring soil health, it's understanding how, the microbial biodiversity or community structure changes in response to management and how we would approach that. Um, but man, those
Mhm.
microbial like measurements, anybody who's done any of that genetic work understands the complexity of just operationalizing those measurements. And we're seeing, the cost of sequencing is coming down and down, so they're more and more available. But that also means that, refining and standardizing those measurements also becomes difficult as those measurements become more complicated. The nice thing about something like organic carbon is it really is relatively simple measurement, right? We ignite the soil and we measure how much carbon comes off of it. And a carbon molecule is the same in Iowa as it is in Nebraska and the same as it is in North Carolina. But there's different treatments of some of those genetic sequencing approaches that can mean those results aren't always as interoperable. So I think there are significant challenges to the genomic side of things to making consistent measurements. And it's going to require quite a bit of investment in R&D, I think, to get good indicators there that are scalable. Not that they can't work in a research environment. We see that in the literature all the time, but
Mhm.
thinking about something that can be applied over millions of acres using commercial labs and commercial sampling infrastructure, I think we're a long way from seeing those kinds of genomic things really bearing fruit in that regard. So yeah,
Yeah.
I'll punt again to the microbiologists in the room.
Thank you. Yeah, and Soil Health Institute is always thinking about scaling, so that's a good way to think about it. And I think we're at time. I see Shannon is here, so Shannon, wrap up this webinar, and thank you again, Jason, for this great presentation and Q&A.
Do you want me to advance to these slides, Shannon?
You're muted, Shannon.
Forgot to unmute myself.
Oh.
Jason, can you just go to the CEU code for now, and then we'll go back through these. That way, if anyone has to jump off, they can get this. Um, Thank you all for tuning in today. This has been a wonderful discussion. Thank you very much, Jason, for taking the time to join us. Please do be sure to check out Farmers for Soil Health, which you can learn more about at farmersforsoilhealth.com. We will have a short Zoom survey once this webinar concludes. So please do provide your feedback on that if you're able to. We do take that into consideration as we are planning our education programs. For those wanting the CEU credit, please open up the CCA app on your phone. Be ready to scan the QR code by selecting Scan Course Code. So we will leave that on here for a second and then we'll go back. We have a couple of other reminders, but I just wanted to be cognizant of the time. So for those seeking the CCA credit for this webinar, the information is on the screen right now. You may scan the QR code on the screen through the CCA app on your mobile device by selecting Scan Course Code, or you can email me, Shannon, using the contact information provided in the chat. So I think that's probably going to be my email. I do see that that is in the chat now. So please get that to us in the next few days or today. If you are if you don't scan the code, please do send me an email. The sooner the better, and I'll get those all submitted as well. So we'll leave that on for a second, and then Jason, if you can, give me a couple more seconds here, and then we'll go back. The next slide we're going to look at, Jason, for your reference, and I think we can probably go there now. We do have, this is the first in our two-part webinar series. So Dr. Jason Ackerson is going to be joining us again next week. So part two is going to be on Thursday, March 5 at this same time. And in that discussion, Jason will be discussing soil health benchmarking, which is an approach to assess how healthy a soil is today and how it can improve over time. So tell all your friends how good this webinar was and that they should join us next week. And, hopefully we'll have the recording out to you before that time. And please keep a lookout for follow-up email with the webinar recording and additional resources. If you have any other questions, please feel free to reach out. Our contact info is on the screen. So Jason, that's the next slide we'll need. So we'll just leave this up here for a minute. And to those that are still with us, thanks. Sorry that we didn't get to answer all of the questions today. This was a fabulous discussion and gives us some insight on what we can include in our future programming. All right, Jason, if you want to go to the very end slide. Thank you again, everyone, and have a great rest of your day.